In order to appreciate the mechanism of action of medications used in the treatment of Cushing's disease, it is important to first understand basic physiology of the hypothalamic pituitary adrenal axis. Have you ever wondered why certain medications provide a more potent therapeutic effect in Cushing's disease compared to others? We will cover this shortly. The hypothalamic pituitary adrenal axis controls cortisol production through a complex system of stimulatory and inhibitory mechanisms. Corticotropin-releasing hormone, also known as CRH, produced in the paraventricular nucleus of the hypothalamus, travels via the hypophyseal portal system to reach corticotroph cells in the anterior pituitary gland. There, it binds to CRH receptor type 1, triggering the release of adrenocorticotrophic hormone, or ACTH, from these cells. Did you also know that arginine vasopressin, or AVP, also known as antidiuretic hormone, a hormone released from the paraventricular nucleus, also binds to specific V1b receptors on corticotrophs? This stimulatory pathway enhances CRH's effects in the anterior pituitary gland, further stimulating ACTH production. Conversely, dopamine, which is released from the hypothalamus, inhibits ACTH release by activating D2 receptors on corticotroph cells. The release of CRH is influenced by several factors: catecholamines, angiotensin II, serotonin, stress, and cytokines, which stimulate CRH release, while GABA inhibits it. CRH not only triggers the immediate release of ACTH, but also increases the expression of pro-opiomelanocortin gene, or POMC, leading to sustained ACTH production. Now that we know the basic stimulatory and inhibitory factors at the level of the pituitary gland, let's proceed to the adrenal cortex. So, at the level of the adrenal gland, ACTH binds to melanocortin 2 receptors present on cells in the zona fasciculata of the adrenal cortex. This stimulates the conversion of cholesterol into cortisol. Cortisol, in turn, provides negative feedback by suppressing the release of CRH and AVP in the hypothalamus, as well as ACTH in the pituitary gland. This feedback loop helps maintain hormonal balance. So, have you ever wondered the relevance of somatostatin analogs in Cushing's disease? Well, both normal and abnormal corticotroph cells in the pituitary gland have two types of somatostatin receptors, SSR2 and SSR5. Somatostatin, a hormone produced in the hypothalamus, works to reduce ACTH production, which ultimately lowers cortisol levels. However, SSR2 receptors are more sensitive to cortisol's negative feedback signals and can be easily suppressed, whilst SSR5 receptors are less sensitive. Because of this difference, treatments targeting SSR2 receptors like octreotide are less likely to be effective in managing conditions like Cushing's disease. Instead, medications that target SSR5 receptors, such as pasireotide, are often more successful in controlling the disease. Pasireotide is a medication that mimics the effects of somatostatin and targets 4 out of the 5 somatostatin receptor subtypes. These are SSR1, SSR2, SSR3, and SSR5. It binds most strongly to the SSR5 receptor, which explains its effectiveness in treating Cushing's disease. Tumors in the anterior pituitary gland, which cause the overproduction of cortisol in Cushing's disease, tend to have more SSR5 receptors compared to other somatostatin receptor types. Cortisol's natural feedback mechanism suppresses the expression of somatostatin receptors, with SSR2 being more affected than SSR5. Because pasireotide specifically targets SSR5 receptors, it is particularly well suited as a treatment option for managing Cushing's disease. As you may recall earlier, dopamine released from the hypothalamus inhibits the release of ACTH by corticotrophs in the anterior pituitary gland. More importantly, about 80% of corticotroph adenomas have D2 receptors, but these receptors are present in very low numbers, which makes dopamine agonists less effective as a treatment option. Medications like bromocriptine and cabergoline, which work by binding to D2 receptors on corticotroph cells, help to reduce cortisol production. However, they are not as effective as somatostatin analogs in reducing the high levels of cortisol seen in Cushing's disease. Now, let's discuss the role of retinoic acid in the treatment of Cushing's disease. In normal corticotroph cells, the POMC promoter gene or pro-opiomelanocortin gene plays a key role in producing POMC, which eventually leads to the secretion of ACTH, as you may already know. Under normal conditions, transcription factors like activator protein 1 or AP1 and nuclear receptor 77 activate the POMC promoter gene. Retinoic acid, when bound to its nuclear receptors, suppresses the expression of AP1 and nuclear receptor 77, effectively blocking the activation of the POMC promoter gene. However, a protein called chicken oval albumin upstream promoter transcription factor 1, oh my goodness, that's a mouthful, or CUP TF1, protects AP1 and nuclear receptor 77 from being inactivated by retinoic acid. In some corticotroph tumors, CUP TF1 expression is reduced, leaving these cells more vulnerable to retinoic acid's inhibitory effects. This makes retinoic acid a potentially effective treatment option for managing Cushing's disease. So what are some of the other effects of retinoic acid? Well, retinoic acid lowers cortisol levels in individuals with Cushing's disease through multiple mechanisms. It reduces the production of ACTH and POMC in corticotroph cells, thereby decreasing the signals that drive cortisol production. We just discussed that. Additionally, retinoic acid has direct tumor killing effects or tumoricidal effects in targeting and damaging corticotroph tumor cells. It also inhibits the growth of overactive adrenal cells, leading to control of excessive cortisol release. Furthermore, retinoic acid downregulates the expression of melanocortin-2 receptors on adrenal cells, further suppressing cortisol synthesis. These combined actions make retinoic acid a promising therapeutic option for managing Cushing's disease. How about medications specifically targeting steroidogenesis? Well, STAR, or steroidogenic acute regulatory protein, plays a key role in adrenal hormone production by transporting cholesterol from the outer to the inner mitochondrial membrane. Once inside the mitochondrion, cholesterol is converted into pregnenolone by cytochrome P450 side chain cleavage enzyme. This step, which occurs in the inner mitochondrial membrane, is the rate-limiting process in adrenal steroid hormone production. Both ACTH and luteinizing hormone stimulate STAR's activity and its downstream effects. In the adrenal cortex, pregnenolone follows different pathways depending on the specific zone. So let's look at zona glomerulosa, for example. Pregnenolone is converted into progesterone by the enzyme 3-beta hydroxysteroid dehydrogenase. Progesterone then undergoes enzymatic changes involving 21-hydroxylase and aldosterone synthase to produce aldosterone, a hormone that we'll review later is very important for salt and water balance. The zona fasciculata, in this zone, pregnenolone is first hydroxylated by the enzyme 17-alpha hydroxylase into 17-hydroxypregnenolone. This compound is then processed by 3-beta-hydroxysteroid dehydrogenase and 21-hydroxylase and finally 11-beta-hydroxylase to produce cortisol, a hormone involved in stress response and metabolism. In the zona reticularis, in this zone, 17-alpha-hydroxylase and 3-beta-hydroxysteroid dehydrogenase facilitate the production of androgen precursors such as dehydroepiandrosterone or DHEA and androstenedione, which are important sex hormones. The mechanisms of action for meteropone, mitotane, ketoconazole, and the newly approved steroidogenesis inhibitor osilodrostat will be reviewed next. So let's look at meteropone. Meteropone contains a pyridine group that interferes with the enzyme 11-beta-hydroxylase. This enzyme, as you may recall, is crucial for the final step in cortisol synthesis. Additionally, meteropone inhibits other steroidogenic enzymes including 17-alpha-hydroxylase and the steroidogenic chain cleavage complex. These effects impact hormone production in both the adrenal cortex and the gonads. Osilodrostat blocks the activity of 11-beta-hydroxylase in the adrenal cortex, but it is more potent and has a shorter half-life compared to meteropone. This allows it to be taken less frequently, typically twice a day instead of 4 times a day for meteropone, and osilodrostat also has minor inhibitory effects on enzymes like 17-alpha-hydroxylase and 18-hydroxylase, although these effects are less significant. How about mitotane? Mitotane has both adrenolytic, causing adrenal cell death, and adrenostatic, meaning inhibiting enzyme activity, effects. It is a chemotherapeutic agent. It has a unique structure that induces mitochondrial damage, leading to cell lysis and necrosis. Its adrenostatic action stems from its ability to inhibit enzymes such as the side chain cleavage complex, 11-beta-hydroxylase, and 3-beta-hydroxysteroid dehydrogenase, which ultimately disrupts cortisol production. And finally, we take a look at ketoconazole. Well, ketoconazole, primarily known for its antifungal properties due to that imidazole group that it has, also inhibits several key enzymes involved in steroidogenesis. Notably, it blocks the side chain cleavage enzyme, which is essential for cortisol synthesis, as we mentioned earlier, as well as other enzymes in the steroidogenesis pathway. These drugs target various stages of cortisol production, offering therapeutic options for patients with Cushing's syndrome or even Cushing's disease. I hope this has been helpful. Join me on our next video where we'll look deeper into Cushing's disease and other endocrine conditions. Thanks for watching.